All right, so today what we're going to do is we're going to be talking about something a little bit different. We're going to kind of move from our conversation about DNA to talking about how we turn DNA into proteins. And we do it through something called the central dogma. Central dogma, another word for that, is protein synthesis. But this is what our lesson is going to be about today, so let's get started. So if we want to remember a little bit from last class, we talked about the differences between DNA and RNA. And we specifically talked about three big differences. We said that DNA was double-stranded while RNA was single-stranded. We said that DNA stood for deoxyribose sugar while RNA stood for ribose sugar. And we said that DNA contained the base thymine, meaning kind of A, T, C, G, what we're used to. However, we talked about RNA doing something a little different, and it's probably the most important difference for today's lesson. We see that RNA actually contains, instead of thymine, it contains the base uracil, making RNA AUCG as opposed to ATCG in DNA. So just keep that in mind as we kind of move through today's lesson. So we kind of start with the word central. We see that the word central means main or important, and we see the word dogma, meaning belief or principle. So this is kind of the main principle of molecular biology. How do we turn DNA into proteins, and how do then proteins kind of make up the rest of our body? So, main belief of molecular biology, just like we just talked about, we see that the flow of genetic information in cells is from DNA to RNA to ribosomes and then to protein. And it kind of involves two main steps, transcription and translation. We talked about this a little bit before we started this video, but just to reiterate that the idea of this lesson is we're talking about how DNA goes from DNA to RNA, and then how we turn the RNA to the ribosomes, and how the ribosomes are then able to churn out protein. And we're going to talk about two steps. We're going to talk about transcription today, and we're going to talk about translation on Wednesday. All right, so just like I mentioned today, specifically about transcription. So how do we turn DNA into RNA? So what is transcription? I mean, we kind of heard the word before, but obviously transcription kind of sounds like the word transcribe, and that's exactly what we're doing. We're transcribing the information in DNA into RNA, because RNA is essentially what is going to leave the nucleus and tell the ribosomes what types of proteins to make. And to transcribe something means to copy it in a different way. An example down here is kind of this scratch paper type writing, and we're able to transcribe it into these lyrics that are very clear. So we remember back when we learned about where DNA is stored, and we know that DNA is stored inside the nucleus, and it can't leave the nucleus. Just like it says, help, I can't get out. DNA can never leave the nucleus. Well, if we need DNA's instruction to make proteins, how are we going to get the information if DNA is never allowed to leave kind of this little shape down here? Well, that's the basis of today's lesson. DNA needs to get transcribed into RNA so that it can then leave the nucleus. So, just like it says here, DNA can't leave the nucleus. How do we get a message to ribosomes? So we need to get this information over here. But we're asking, how do we do it if DNA right here can never leave this little bubble? Well, we use something called mRNA. M meaning messenger RNA, and that's exactly what it's doing. It's acting as a messenger for DNA. So today we're going to talk a little bit about how we do that. The process is actually quite simple, and it shouldn't take us no time at all to get through it. So step one, just like we had DNA polymerase, and DNA polymerase's job was to add those nucleotides, those A's, those T's, those C's, and G's to a template strand to copy DNA. And RNA, obviously, we have RNA polymerase. And the job of RNA polymerase is to unwind DNA, just like it's done in this picture. And it unwinds it so that then now you have two strands that are exposed. In the next step, what RNA polymerase is going to do is it's going to add nucleotides from the 5' to 3' direction. The 5' to 3' direction is not super important for the purposes of our conversation, but just nice to know. Remember the rules. So in DNA, we would have just paired these up. We would have gone C's to G's, G's to C's, A's to T's, G's to C's. However, remember this exception to the base pair rule. Remember when we talked about the difference between DNA and RNA? In RNA, we use the letter U instead of the letter T. So as we kind of pair these up, we need to make sure that we're not including any T's when we are creating mRNA. So as you've seen that dRNA, what RNA polymerase has done is it's actually made a copy of the strand in accordance to, obviously over here, it's made a strand of mRNA. And it's going to pair C's still with G's and G's still with C's. It's going to pair T's still with A's, because remember, DNA still has T's. T's are still going to pair with A's. However, whenever we see an A in a DNA strand, what the mRNA strand is going to read is a U instead. What happens afterwards is DNA is essentially going to rewind itself back up once mRNA has been created, and mRNA goes and leaves the nucleus so that it can do whatever job it needs to be done. So here's an example that I'm going to give you, and this is just a quick example, and then we will move on to some practice. So remember that RNA is A equals U and U equals A, and G equals C and C equals G. Please remember that. So if you look at this DNA strand, what do we think is going to happen? What do we think A is going to pair with? Well, normally in DNA it would pair with T. But today what we're doing is we're taking DNA and we're turning it into RNA, mRNA, so that mRNA can then need the nucleus. So if I ask you to write an mRNA strand, instead of putting T here, you should actually remember that a U should be matched with A, because A equals U in RNA. C is still going to equal G. T is still going to equal A, because remember, in DNA, T exists, and T in RNA is going to pair with A still, because A is still a nitrogen base. So essentially what we're doing is we're turning all A's into U's, however, T's still pair up with A's. So I'm going to go ahead and show you what that would look like. So as you can see here, A pairs with U. T is still going to pair with A, because if you look at this mRNA strand, you have to still have A's in them. T is not going to pair with U. T still pairs with A, because A is still a letter that is probable in RNA. We're going to now move on to some practice. So hopefully this lecture made